Think forward. Think Research Channel.
Beyond the neon lights and gated communities of Las Vegas lies the Great Basin Desert, a large, sparsely populated area covering most of the state of Nevada. The region is marked by isolated mountain ranges, arid valleys, and very, very hot springs. Although they look inviting, many of these springs are far too hot for humans to enjoy, with temperatures near or above 160 degrees Fahrenheit. When water temperatures are this high, photosynthesis, the process by which light is converted into chemical energy, cannot occur.
This is a case where...
And that's exactly what intrigues microbiologist Brian Hedlund. The UNLV assistant professor studies microorganisms that thrive where other life would perish, in the very hot springs of the Great Basin.
At high temperatures, at some point you get up enough, it's about 163 Fahrenheit or 73 Celsius, photosynthesis doesn't occur anymore. We're not exactly sure why. Life then has to work in a way, in certain ways, that are separate from photosynthesis. These reactions philosophically might be similar to the ones that occurred before photosynthesis ever happened on the planet.
The way life occurs in hot springs is through a process called chemolithotrophy. In this process, microorganisms like bacteria feast on inorganic substances like methane or sulfur for the energy they need to survive. Hedlund has measured many of the inorganic substances in the springs and uses these measurements to predict the types of chemicals microorganisms use for energy. But the task is far from complete.
These bar graphs represent diversity of the organisms that live in the hot springs, and these bar graphs show what groups of life they belong to, and these slate gray colors represent groups of life that are unknown. We have a gene sequence for these organisms, and that's all we know them by. We don't know what they're doing, we don't know how they're functioning within the hot spring. We really have no idea what they even look like underneath a microscope.
The goal is to identify the forms of life living in the hot springs, predict how they derive the energy they need to sustain life, and then test those predictions.
What we do is take some chemical measurements out in the field and do some chemical calculations to see what the organisms can be eating, what they can be breathing, and how much energy they get from these reactions.
Potential food sources in hot springs include hydrogen, methane, sulfur, and magnetite, a mineral found in magnets, while chemical compounds such as oxygen, nitrate, or nitrite are common substrates for respiration.
We throw all of our measurements from the field into an equation to figure out how much energy is available when you mix and match these different food sources and respiration sources for the microbes.
In 2006, Hedlund was awarded a 5-year, $840,000 grant from the National Science Foundation. He used some of these funds to take a team of colleagues and students on a 6-day scientific field trip to Gerlach, Nevada. The trip had several goals. The first was to collect samples and measure chemicals in the Great Basin hot springs for later analysis in the lab. The second, to collect DNA from cells and viruses for sequencing. Third, to collect samples to cultivate new microorganisms in the lab. The fourth was to quantify the microorganisms in spring water, and the fifth was to introduce basic concepts of microbiology to students attending Pyramid Lake High School.
My grant involves not only UNLV students, but also students from Pyramid Lake High School. And this is on the Pyramid Lake Paiute Reservation in the town of Nixon, Nevada. I'm hoping that in the process of focusing on the science of these hot springs, we can also talk about the history of the importance of hot springs to the Paiute people and how they use them and why they use them and how they think about them and how they feel.
Pyramid Lake High School is located in the heart of the Pyramid Lake Paiute Tribe Reservation, just down the road from the lake for which the reservation is named. Although estimates vary, about 1,700 northern Paiute people call the reservation home.
The tribe basically realized, hey, we need to start our own school. There's things that we want to accomplish, and one of them was like having a Native Studies program in their school, and that would include like the language, the history, and the arts and crafts.
NSF has this research focus. So this is just a little bit of a diagram of what a cell is. And so we have to talk about this when we talk about microbes. But like I said, they also have an educational focus. Okay, they've decided that our science alone is not enough. If I learn something and nobody knows about it, who cares, right? We have to marry the science, the progression of science, with the progression of educating people on the way we do science and what we learn. So what do I do? So you're going to go, you want to streak, so I would hold it like this.
There is a cultural tie. Some of the kids that have been out here haven't been able to see some of these areas that were deemed traditional Northern Paiute territory, but now we're out in the field and they're able to experience that.
I think I do. What do we do with it? Just do the same thing?
Yeah, just like you did with the photobacterium in the lab. This won't have that many bacteria on them, so you could streak them, a little bit more wildly. It'd probably be okay. Yeah. Or maybe better.
All right, I got one.
The Gerlach Hot Springs are located on private property just outside the tiny town of Gerlach. With the permission of landowner Dave Jameson, Hedlund and his team set out for their first day of chemistry and sampling.
This is kind of the spring that we focus on the most. We have a numbering system. We call this GBS 17A. That one right there, behind it, is probably below 73, and we can go and have a look at that one. This one that's connected behind, people actually swim in that one, so that's in the probably maybe 40 or something like that. And this one was named by John C. Fremont himself in 1844 or so, Great Boiling Spring. So this is the spring that the complex is named after. So this is the hottest we've ever measured, this one. I'm up to almost 86 Celsius in here.
You can't? Just look up.
We're going to be measuring the chemistry. All the Lamont kit chemistry stuff will be in the tent. So we can leave that, why don't we leave that in the tent, and we can just log as we go. No, carry that with you, yeah.
Okay.
Okay, and then we'll transfer this into that. I'll get you started. We'll make sure everything's running okay. And I'll probably try to do a whole one with you. And then I'm going to be running around. We have different chemistry teams out there measuring different things. What we're trying to make sure right now is that each team is actually competent enough to get going through the springs in a way that we can be confident of some data in the end. But there's this teaching mission that's constantly going on, and it's fairly complicated right now. So I'm mildly frustrated. so we're learning here. This marker is really important because everybody doing every chemical measurement is going to follow us and log it this way. Okay. All right? So that's going to tell everybody, take a sample here and label it this way. So I'm going to have to give a speech many times here. So I need more, I know everything and I need to get it out to everybody. The most sensitive stuff is under Tristy's control, and we've talked about it. She's done it before. It's kitted, everything's washed very carefully. And so I'm very confident of that. And what I'm not confident about right now are these individual chemical measurements that individual groups are doing.
If you want, yeah, just keep chugging through this list and then I'll start on this one.
We have a spreadsheet of all of our data for the last few years. We've got that with us. Robin is actually in charge of keeping the master notebook for the trip. So we'll be able to, with just a click of a page in the spreadsheet, compare last year's data to this year's data. If the worst thing we have to do is come back up here, we could do it. We come up here periodically for, specific experiments anyways. But I really don't want that to happen.
It's day 2 in the field, and the scientists have settled into a routine. On the agenda today, tying up loose ends from day 1, completing special projects, and preparing for another day with high school students. One of the special projects for the day is to capture a large number of cells from the springs. Tristy Vick and Jeremy Dodsworth teamed up with Kui Wang and Steve Techman from the University of Maryland. The goal? To employ a special tangential flow filter to capture the cells. These cells will be used later for DNA sequencing.
If we get enough DNA, then we can sequence it and get what's called the metagenome of the community. If we can get this to work and we can get enough DNA and get this done, I think it's a really big deal. Nobody has done this exact thing on hot springs before, that this would be kind of a new type of community that nobody's really looked at this way. So I think it's really, really exciting.
Other teams stayed busy tying up loose ends and working on other special projects.
Today we're going to be working on trying to cultivate some of the microbes out of our flagship spring, Great Boiling Spring 17A. So we've designed a background medium. We'll add some reagents for the microbes for food and for some sort of source for them to have something to breathe. And then we'll inoculate the tubes and leave them sitting in the hot spring so they're incubating at the right temperature, and come back in a couple days and see if anything grew.
I would try first just kind of teasing one of these things onto a slide as opposed to pipetting. We can always do that if we have to. I would just go there. We've got a lot of material. That's a lot though. That we won't be able to see light through that. I mean, if you can see any material, it's going to be a ton under the microscope. Yeah, I mean, this is going to be loaded.
We're kind of documenting exactly where we sampled for, in case we come back and want to sample the same area. So we're taking a picture of the stake so we know what spring it is, and then a picture of the exact sample site where we took it, and then a picture of the whole spring. Just in case there's any doubt later, we have at least the record to go back to.
So now we're making a slide for the microscope to look at. What we're going to do is take one of their samples that they took and gave to us and put a bit of it under the microscope on the glass slide. We'll probably make a few more later when the kids come.
You want to talk about our petri dishes first? Ooh.
That one's a dog.
Dog. Dog mouth. My mouth. Oh, fantastic. Actually, this is a little bit interesting. It's not generally very easy to look at these colonies and tell what the organisms are, but this one, I might have some guesses. This one is interesting. There's one that has a darker center and it's almost a little bit fuzzy looking. It might be a fungus, but I think it's probably an actinobacterium. And that is the group that makes a lot of antibiotics. So a lot of companies are interested in growing this type of microbe and then screening it for activities, for antimicrobial activities that kill other bacteria.
I really like the fact that we've been helping out with the Paiute kids. Mel, the kid I was working with yesterday, was great, and he really got into it, and he's like an expert at measuring sulfide now.
This is like a lot of stuff that I didn't know about, like all the microbes and everything, and the hot springs. I didn't know a lot about the hot springs.
This is very good for me because I can come out and learn a lot of stuff that I never knew and see all the little bacteria or stuff that's very small for us to see with our own eyes, and we can see under a microscope.
I'm hoping that this kind of motivates them to think about what they're going to do after high school. All in all, I think this is good for them, and it's good for us because we get what we want and they get something out of it too.
Who wants to see those streamers we were just pointing at and looking at? Our DNA work tells us that there are many, many microbes in here that are new phyla of microbes. They're not related to anything we know, but I don't know what they're doing or how these correspond to our DNA sequences.
So we've been in the tent all day getting our media ready for growth, and we've inoculated everything. Now we're trying to find a spot in the hot spring where we can leave these tubes to incubate at the right temperature. So we'll leave them in the hot spring for about a week to see what grows in each of the tubes.
Because we were out here yesterday, we're a little bit more familiar with where all the springs are and what we actually want to accomplish out here. So today is a lot easier, and it seems to be a lot cooler as well. Overall, we're getting a lot of good results, but we'll see when we get back to the lab.
All right.
Ready?
3 more to go, I think.
Let's see.
That one barely has a scratch in the ground.
Hedlund and his team had a few more samples to collect before leaving for the second leg of the trip in Surprise Valley, California. But before the work was begun, Hedlund surprised his team with a short visit to the privately owned fly ranch, just north of the hot springs and home to this remarkable site.
This site is very interesting to the public because it's beautiful and it's a habitat that's so extreme that there is no macrobiology. This is all microbiology, all geology, and all chemistry happening right in front of us. So it gives us a unique opportunity to study life and the absence of plants and animals, which is, as we understand it, more representative of the long-term history of life on our planet. So when I see a place like this, I, in a way, think of looking back in time toward a more ancient life system on Earth. It's like sitting at a table and having a lazy Susan constantly delivering food. You know, just coming right by you. I'm hungry, I'm hungry, I'm hungry, food, 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 light, light, light.
Yeah, it's cool.
Just being bathed in food, coming right by, 24/7, 24-hour buffet.
How long are we going to be here?
Not long.
Oh, just to get ice?
Actually, I don't know. I really don't know. Greetings. I think the plan for today, Kelsey and I have some quick sampling to do. I propose that we'll do that as fast as we can, and while we're doing that, Robin, why don't you lead people over to Sandy's, and we can, with the temperature probe, kind of lay out the other sampling sites. Then I estimate maybe sometime around 2, something like that, we'll head off for Surprise Valley. We're looking for certain groups of microorganisms or certain processes by looking for genes for those processes through temperature. What we're going to do is lay out a sampling point at 70, 60, 50, and then 40 if we can. We'll take the chemistry so we can get an idea of how the chemistry changes, and then we'll take samples that later we can use for DNA analysis.
59.9.
Perfect.
60.
Perfect. That's as well.
Staying.
Staying?
Yeah.
Perfect. All right, this is it. This is the second spot. Still a little high.
55.
Too hot. Let's see if we can at least knock out a 50. Still going up. Try a little bit further. All right. This is it. So we were looking for a 40-degree sample, and we didn't find it. So we'll have, 80, 70, 60, 50. It's a good thing we put the protocols in all these copies, because the master protocol for sulfide and oxygen is somewhere. Yeah, probably with Cameron, who's on his way to Reno right now.
Where's he going to Reno?
I'm going to Reno to use some equipment at the Desert Research Institute. So we're going to, you saw all that cell concentrate from the filtration.
Yeah.
They're going to go down there and spin that in a centrifuge. So it's going to be spinning it really hard with lots of force. So that force is going to push the cells into a pellet in the edges of the tube. And then we're going to freeze those pellets, and we're going to pour off the supernatant, which is the liquid on top. that's going to contain the viruses. So we'll send those viral samples off to my friends at Wisconsin and keep the cells for ourselves. You guys are all set? Chris is the sample committee?
There's nothing that we can do until you're done. Because we have to do sediment.
You can, this one for sure, you can take, what we're doing isn't sensitive. So don't worry about us. Chris grabbing the bottles is.
Yeah. So you want us to go and follow him?
The good thing is there's a
settled by the time you come back for hydrogen sulfide?
Yeah. Hydrogen sulfide we're going to have to do way later because there's only one color emitter and that's going to be over there with everybody else. So that's going to be kind of a scramble. Maybe Mel and I will skip lunch. We'll just give up lunch for science.
Thanks.
Thanks, Mel.
Okay, so we're going to start from the front and do the sediment sampling, but I think the girls will like that anyway.
Yeah.
Tucked away in the northeastern part of California is Surprise Valley, a remote, high desert area ringed by mountains and bubbling with hot springs. And it's here, at the posh Surprise Valley Hot Springs Resort, where the scientists and high school students convene for a final day of research.
Good. So Gerlach, the hot springs there, we'll go through this a little bit, but they really are microbiologically and chemically unusual, and my work is focusing more on those springs. I see a duty of ours is to spot research around to understand what normal is or what the range of possibilities are in the Great Basin, comparing them to other places, and Surprise Valley is certainly one of those comparative sites.
To make these comparisons, Hedlund and his team repeat the sampling and chemistry measurements they took in Gerlach earlier in the week.
Over in Gerlach, the salt is mainly sodium chloride, so it's table salt. And here, the major cation is still sodium, but the major anion is bicarbonate. So it's sodium bicarbonate. So that over in Gerlach, it's like table salt, and over here, it's like baking soda in the water. This is the outflow from SV2 coming down here, and what we measured 90 or 91 Celsius in there. So this is really hot water coming down, and you can see that, well, maybe at the fringes or the splash zones, there's photosynthesis going on, but the bulk of the spring is too hot for photosynthesis. You can really see what the temperature limits for photosynthesis are, just based on the colors here. So at the high temperature limit, you get this intense chartreuse, and then it'll become green, and then you'll see a more robust community, often with the carotenoids, when it gets a little cooler. So we have a few different springs that kind of look like this, but they're smaller. I'll take you guys to those, and then we can see some other interesting things. It's very, very difficult to have these students with backgrounds that I don't know and abilities that I don't know, and to move them forward. But I think that was the simple goal of the outreach portion. No matter where they were, we wanted to increase the appreciation of the students for science and push them forward no matter where they were. So here's one interesting feature right in here. You guys see that stuff attached to the logs? What does it look like? Hair? Excellent.
You know, it's really great that he really wants everybody to learn something too. You know, it's not like he's just sitting there saying, okay, collect my samples and that's it. You know, he wants us to learn, and he wants these guys to learn.
Microbiologically, one of the biggest things we see here is that these systems have a large presence of bacteria called aquaficae. You can see these organisms in the outflows of the springs especially, and they'll form these hair-like aggregations. Those organisms are probably called Thermochrinus albus, or hot white hair. Here at Surprise Valley, we see a large component of these organisms with our eyes, and also we see them when we do our DNA analyses. Over in Great Boiling Spring, we don't see these things with our eyes, and then we don't see the DNA for the aquafecales when we look. It seems that aquafecales are in fact very important here, but probably not very important over there.
I think he's made a connection with the kids, and what's really invigorating is seeing his excitement with his own research.
I think the high school kids are having a good time too. They're really enjoying coming out here and learning how to do something that they've never seen before. They're really interested in this equipment and what we're doing.
When he first approached me last summer, he told us his goals, and to me, he's achieved them because he did include us in the research.
Well, we definitely wouldn't be here without Brian. He put everything together, he organized everything, and even if leadership isn't completely visible all the time, just the fact that people know that it's there, people need that. It kind of keeps everything going.
I've picked up a lot more skills. Yesterday, even when I went to the Desert Research Institute in Reno, they helped me out with some epifluorescent microscopy using some DAPI staining, but they showed me a new way of doing it. So it was great. I don't think I would have gotten that if I had just stayed in the lab.
Field work is fairly rare for us. It's important for us, but we don't have time and money to always be out here. And it's limited, so every time we come out here we take samples that we analyze for long periods of time.
It's day 6 of the summer field trip, and the scientists and students have come full circle with the return to Pyramid Lake High School. The assignment for today, to engage in a bit of microbial forensics using some of the samples collected during the week.
Basically what we're looking for in these microbial cells is a fingerprint. So just like your fingerprint, someone can look at it and know that it's you because it's unique, but everybody has a fingerprint, so you can compare all of them. So the ribosomal RNA gene is a lot like a fingerprint.
So Tristy's going to bring a rinse over, a washing solution. So we're going to pipette 500 microliters of the washing solution onto the column. Then we'll spin it again.
After all the hard work, everyone is ready to kick back in the refreshing waters of Pyramid Lake.
So here we are at Pyramid Lake. The trip is just about to wind down. And of course we reached a lot of our scientific objectives, but we also reached a lot of our broader impact objectives. All of the high school students got to know us better and got to know our science better, but we also got to know a lot more about them and their culture, and that was really a big goal of ours. And so we're out here having fun, splashing around for a few more minutes, and a little bit after that we're going to pack up and head south.
I'll give it a shot. Toss in. Huh? Just going in.
We'll be analyzing data from this trip probably, for several years into the future. The sky's the limit.